This is episode 109 of the Immunology Podcast, Phagocyte Cell Biology with Dr. Johnathan Canton. Hey everyone, this is Dr. Jason Goldsmith and Dr. Brenda Roud. Welcome back to the Immunology Podcast, where we have conversations with immunologists. The Immunology Podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. If you enjoy the Immunology Podcast, rate us and leave us a review. We're always looking for feedback on how the podcast can be improved and for suggestions on guests. Today, we have Dr. Johnathan Canton from the University of Calgary on the podcast to talk about his research on dendritic cells, macrophages, and innate immunity. We've also got our usual roundup of recent highlights in immunology news coming up. But first,
we are a month away from iOS 2025, and we cannot wait to attend and cover the International Union of Immunological Societies meeting in Vienna, Austria, between August 17th to 22nd. Very excited. So if you want to learn more about iOS 2025, you can check their website at iOS2025.org. We hope to see you all there.
All right, Brenda, have you booked me restaurants for our free nights in Austria yet?
I have not booked the restaurants yet, though. I already have a list of places we should go. But so we have some cafes, because the thing also in Vienna is, the these fancy cafes, where like cappuccino is from or something. So we're going to go get some Sachertorte, some sweets. So I already have those, in my list.
My stomach is trusting you.
Yes. Yeah, we're going to get some Wiener Schnitzel in the place.
Yeah.
I'm so excited. Get some nice wine. It's going to be great. So exciting.
Sausage.
Some sausage as well. Yeah. All the classics.
Excellent.
Yeah.
I'm excited.
Yeah, indeed. So, but while we wait, what do you have for me today?
So I'm going to start with the CAR T cell therapy paper.
All right. Nice.
Because I'm you now. Apparently.
Yeah. Stop stealing my topics, Jason.
Hey,
stay on your lane.
You picked the papers before I did, and you passed on this one. So
That's true.
It's all on you.
It was a tough choice. I did let that one go. So I'm glad that you picked it up. So
you went full Elsa and let it go.
Yeah. Oh, God. Go, please.
So in Nature, coming out July 2nd, we have rewiring endogenous genes in CAR T cells for tumor-restricted payload delivery. So first authors are Amanda Chen and Ka Min Yap, and last authors are Philip K. Darcy and Paul Beavis. All right, so apparently, so we all know CAR T cell therapies had a heck of a time in solid tumors. And partially that's seems to be because the microenvironment is so suppressive, the tumor microenvironment, that you can't get the CAR T cells, even if they're targeting right, to do enough, because they just kind of go in there and then they get sleepy and then they go to sleep and they don't work. So this is how I tell the story here. So there's a concept of armoring T cells, apparently, where you put them under a promoter that should preferentially activate in that tissue microenvironment. And they've done this with IL-12 and IL-2 before, but to date, it's been these promoters driving IL-2 and IL-12. They work on the tumor, but there's a lot of toxicity involved with it because the promoter's not restricted enough. So you're still getting peripheral IL-12 and IL-2 happening, and that's bad news bears. So this group basically did a CRISPR screen. So they did a CRISPR knock-in strategy, but what they did is a general screen for different. So let me take something. They did two things. First, they developed a CRISPR knock-in strategy using PDC1 as the prototype gene because it's increased in T cells on activation, and showed they could put GFP in here or IL-12 or IL-2, and this knock-in approach worked with OT1 cells or regular CAR T cells, and armoring was effective. But they also noticed that it had the toxicity, but they're really validating their CRISPR technology first. So they said, okay, let's go find a better locus. So they did RNA-seq on CD8 CAR T cells isolated from the tumor and spleen in xenogenic Lewis Y or Lay-Y CAR T cell model animals. And so they tried to have a humanized approach, trying to find and really narrowing on promoters, on genes with specific promoters that they could find that had high increases in the tumor, but not in the spleen or liver or other places. And so they really then gated on this very extensively. They found 27 genes with a partial select for 20 analysis based on the magnitude of differential expression, and then 12 from there that were better than PDCD1, and then went from there and did knockouts and looked at what was affected and got top 6 genes, and then from there kept narrowing down, and then looking at tumor-restricted GFP expression as a surrogate. And these promoters, NR4A2 was the main winner, and then RGS16 was also ones that required even more potent intraterminal expression, but it had a little bit more background. So they went with NR4A. They made an NR4A IL-12 armored T cell in the mouse system, again, OT1 and the CAR T cell systems, and it worked beautifully, suppressing tumors without a lot of systemic exposure to IL-12 and other markers of that. They tried that with IL-2 then as well, and that no worky because it needed even more IL-2 than it could drive. So they switched to that other promoter, and it really worked there. And so they then kind of tied this up and said, well, what if we did with the humanized CAR T system? And they did that. And then they looked at some of these markers in human data sets and say, hey, we see these are expressed up in the human data sets too, go team. And I think they're ready to go into clinic now if this is an approach, because they have the data showing they have less exposure and as humanized of a mouse system as you can get. So it was pretty cool. It's really an example of like armoring is a great concept, but they couldn't quite get the specificity. And so they really focused on that.
Yeah. Yeah, it's like I really like how they say we don't have a good promoter. Let's just try to find something that is very distinguishable, very differently expressed between where we don't want this thing to be expressed and where we do want it. I'm kind of surprised that they found such a good candidate that works because, yeah,
Yeah, it was really neat. I like the approach.
no, indeed. And then so IL-12 basically was the best option to armor the car with.
Exactly. Well, it's IL-12 is already what they did. They knew that. That's not what they changed. They put it under a new promoter, so you want to have IL-12 dumping peripherally.
Yeah.
Toxicity.
Yeah, that's always a problem. All right, that's very nice. That's very neat. I like such a, what's the word? Such a nicely logical steps to find. All right. So but why don't we stay on, the tumor immunology field? And I'm going to talk about my first paper today, which is published in Cancer Cell and is titled Subclonal Immune Evasion in Non-Small Cell Lung Cancer. And I have to say, well, first, let me just give some credit to the authors. First author, Krijn Dijkstra, and the corresponding authors are, the senior authors are Sergio Quesada, Emile Wust, and Charles Swanton. This is kind of both from the University College London and Emile Wust from the Netherlands Cancer Institute. And it's a nice, I think it's a very nice study. It's clearly a lot of work. And I think it's most interesting, not necessarily for what it tells, but what it's enabled to tell. So let me elaborate a bit. So we know always tumor immunology, one of the basic, accepted truths of tumors is that they're heterogeneous. And we have within one tumor, we can have areas that are hotter, they have more infiltration and more lymphocytes and other immune cells, and potentially they're being subjected to kind of some kind of immune attack. Then you have tumors that are more cold. So usually that's what is referred to a tumor that does not have infiltrate, that if you do a stain, you don't really see immune cells infiltrating, and the things in between. And another concept is tumoral escape. Sometimes you treat tumors, especially now in the age of immunotherapy. Not everybody responds to immunotherapy. And to some extent, this can be attributed to this heterogeneity. You can imagine that some clonal versions of the tumor cells are more susceptible to the immune response than others. And there's still a lot to understand about how tumors, especially when you see a tumor, right? At any point when you excise a tumor from a patient, this tumor is just a snapshot at a specific time. of a whole history that a tumor had before we actually could be able to detect it or before we noticed the tumor was there. And this is part of this big Tracer X consortium that it's from I think it's a big collaboration in the UK between the Francis Crick Institute and several universities, in which they're trying to collect tumors or material from patients at different times in order to try to understand tumor evolution and how, whether we can predict, if we have a snapshot of a tumor at an early stage, can we understand how this tumor will respond to immunotherapy, how this tumor will kind of evolve in time, and whether also we can, if you see a snapshot of a tumor at a specific time, whether we can, in a way, fill the gaps of what happened to the tumor before, and better, in a way, to better understand what are the weaknesses of, cancer. And so this is part of a huge consortium. They have tons of samples. They're following patients across time, before treatment, after treatment, if they have a relapse, and they just take samples. Sometimes I think they're also doing postmortem for those who unfortunately don't respond well to the therapy, and they're really trying to understand. So one of the things is the concept of immune evasion. Why are tumors escaping the immune response, right? Because that's why we see them. If they didn't escape the immune response, they wouldn't be there. We would never see them. And so are there things that are intrinsic to the tumor cell that are preventing the recognition? Are things about the environment? Are things about the T cell repertoire? So in this study, they try to look a bit better into how does the heterogeneity of a tumor impact the immunogenicity. And I think, in a way, you say, well, I mean, maybe they're losing expression of MHC molecules. Maybe they are losing the gene that is being recognized by the T cells in this tumor. And I think it's interesting because actually they cannot see in this study, they actually don't see these mechanisms as important. So they still do all this work and they're still not sure. They cannot explain why some of the clones are more immunogenic than others. So let's dive into the data. Basically, the first part is that they take a lot of patients with lung cancer, and they have. So what I can imagine was a huge work. The tour de force. They generate organoids derived from these tumors, but not just from the whole tumor. They do they try to separate different regions of the tumor. So they generate organoids derived from different regions. And then they also make clonal organoids from each of those regions. They make several clones. So they pick up several organoid clones to have homogeneous tumor cells in a way that they can kind of be representative of the heterogeneity of the original tumor in the patient. And I can imagine this was a ton of work. And although these people have a lot of experience making organoids, it's really hard. And I think they mentioned that they started with like 60 patients or something around those numbers. And they could only really generate a few organoid lines. So that was already, I can imagine, a huge hurdle in this work. They started with 42 patients, and they could only establish from 7 of them, it could actually get organoids to work. And only for 4 patients, you could actually make organoids from multiple regions. So can you imagine this is a very difficult thing to do. So basically, most of the work is looking into the organoids derived from 3 patients. And I would say most of the interesting data comes from 2 patients in which they could do the organoids from several regions, so many clones from different regions, and also expand tumor infiltrating lymphocytes to see what is the repertoire of the T cells that are infiltrating these tumors. And basically, what do they see? What are the take-home messages from this work? What they show is that indeed they make this organoids and they look into the repertoires and they find differences. Basically, look at the 2 patients in which they have different many organoids made. They try to understand why some of the clones. So if they co-culture the organoids with the expanded tumor infiltrating lymphocytes from for the same patient, they see that very distinctively some organoids are susceptible to these T cells, and some organoids are not. And they're all derived from the same tumor, right? From the same patient. And they see that there's differences in the T cell repertoire between the two patients. One of the patients, interestingly, has a very dominant T cell clone. But then basically because they're focusing on the organoid part, they say, well, some of the organoids are attacked by these, are recognized by these T cells, and some are not. So maybe it's just that the organoids are inhibiting the T cells in a way. But then if they are loading the organoids with peptides and they're putting T cells are recognizing these peptides because they are transducing a TCR, for example, they see that there's not a problem. So it's not that the organoids are unable to present antigen or they don't have MHC, because if they put artificially, they add an antigen, then the T cells can recognize these organoids. They also look into, are they losing MHC expression? That's not the case. Are they losing expression of, no, they do a whole exon sequencing, and they don't find any differences between hot and cold clones of gene expression, of mutations that would explain the loss of maybe antigens. They don't find differences as in antigen presentation. So in the end, you say, well, maybe it's unclear why one organoid will be recognized by the T cells and the other won't, and why are these organoids being inhibiting the response by the T cells differently. And it's not explained by loss of T cell repertoire. So it's very interesting to see that. I thought this would be a very kind of straightforward. I think that probably that's how they started this project. It would be a very straightforward show of mechanisms of immune editing. But in the end, the conclusion is that it's still, they still cannot show where does this different immunogenicity come from, even though they have individual clones of the cancers and they have the T cell repertoires of these patients, and they still cannot explain where this is coming from. And I think it's very interesting because I really thought this would be this would have been the perfect system to show this, but it's not.
That is super fascinating.
Yeah, so I guess this really show how hard this is, but I think it does make the whole platform that they established, generating the organoids and studying the T cell repertoires, I think is very valuable. And I'm really surprised it doesn't show more about where does the immune evasion come from. But I think we should still be doing experience like this, because even if it's a negative result, it does it does show that this might be a way of starting to find which these mechanisms are, because you could still see the differences in the immune response. I also want to mention that they also look a bit into NK responses, and again, they also see differences in NK. For one of the patients, they couldn't get culture of expanded T cells, so they couldn't look into an antigen-specific T cells, but they look into NK responses, and again, they see a lot of heterogeneity. Some of the clones are able to induce T cell NK cell activation, and other ones don't. But it's again not clear what is behind this different recognition.
Sounds like some people have some work left out for them.
Yeah.
Which is good. It means not everything's solved yet.
Yeah.
All right. Well, I don't have a segue here, so I'm just going to jump in because I can't even come up with one for this. This is also in Nature, July 2nd, titled WSTF, Nuclear Autophagy Regulates Chronic but Not Acute Inflammation. First authors, Yu Wang, and corresponding authors are Zhao Wang, Terje Johansson, and Zhizhuan Dao. All right. So, in this study, we know that autophagy regulates all types of stuff, but usually cytoplasmic autophagy. And this is looking at what is happening at, it's kind of an interesting mechanism. So they have this WSTF, which is a chromatin binding protein. And during chronic inflammation, they find out that it undergoes autophagy by being exported from the nucleus and then being degraded. That autophagy, when it's gone, opens the chromatin up, allowing for transcription of chronic inflammatory genes separate from acute inflammatory genes. So this is really a chronic process. So if they look at acute inflammation, it doesn't work. If they use MASH, so metabolic dysfunction associated fatty liver disease, steatohepatitis, and the osteoarthritis as two mechanisms. But it's fascinating because you're having this whole other regulation going on. So they look for nuclear autophagy substrates, doing a screening. They do kind of a IP pull down with tandem mass spec, do a whole bunch of gating on that, and WSTF comes up, and then they try to understand what's promoting that and figure out that ATM, which is one of the that is present in senescence, drives it. And then they get into a lot of mechanistic weeds, which I don't want to go into here because it's just a list of things that bind. But the point is that they do map out the pathways, and WSTF has an ATG8, which is one of the main autophagy proteins, part of the main molecular processes. And so it is degraded through ATG8 interaction. Okay, so they figure out when you lose this in vivo through knockout systems in mice and in cells, you pump out more inflammatory cytokines. They then go into acute versus chronic models of signaling and look at that acute inflammation doesn't do this, but chronic inflammation has this autophagy-driven response that then leads to this degradation. And that's kind of the cool things, that autophagy is driving chronic inflammation. Then they design peptides that stabilize WSTF by blocking the interaction for autophagy. And so this is a sink. And so, these little mini peptides get sucked up for autophagy. And so they make some peptides that target in, and then have even ones that are designed that you can just put on, in a Petri dish or into mice, and they get integrated further. So they'll go into the cytosol and then have nuclear transport based on, those little sequences you put at the end. And so they build these, and then they show that, this WSTF signaling process goes away, right? So that you have less chronic inflammatory cytokines in a chronic inflammatory state if you dope the protein in. So they have cell penetrating peptides, and then they go into the murine models of MASH and show that if you can inject this peptide, it works to prevent inflammation in MASH. And then they extend this out into arthritis models as well. And so I think there's a couple cool things. One, they're providing a therapy that treats it by blocking this. And that's really awesome. Two, they really linked that, whereas we think about autophagy being kind of a acute inflammation, not necessarily acute inflammation, but kind of a different regulatory process, you're really saying that autophagy is triggered in chronic inflammation, and that's degrading nuclear autophagy, right? Cytoplasmic autophagy is old news. Nuclear autophagy. So signals that export proteins to the nucleus for autophagy is then opening up the chromatin, leading to the chronic inflammatory state. And there's a very clear linkage for a bunch of the cytokines in chronic inflammation that they're talking about here that is driven by this. And they have then the therapy to make it work. So I thought that was pretty cool. Not a signaling mechanism. I've done autophagy work before, but it's all been the cytoplasmic variant, not autophagy of nuclear proteins. So I thought that was really neat. They spent a lot of time validating their IP because a lot of co-immunoprecipitation excludes the nucleus just when you spin it, right? And so they say, no, we're really collecting the nucleus here and looking. So I forgot to mention that, but that's a key thing, is they went fishing for the nuclear proteins and had to modify the standard protocols, which is why people haven't seen this before, is because the standard protocols exclude that entire pile. So there you go.
That's funny. Yeah, that's very interesting because it's true. When I think of autophagy, I think more of like non-inflammatory, like I think of anti-inflammatory conditions. I don't know, like I don't think of inflammation and autophagy together. But then I guess, yeah, I was thinking of like the regular cytosolic autophagy, not nuclear. Um,
Yeah, well, that was it.
okay. Are they planning to do a clinical trial or something with this? Can these peptides be
Probably, but they don't
ruled?
they don't flag that.
Yeah, that's very interesting. All right. So for the last paper of today, we're gonna stay in the liver. It's nice. We've had a little bit of a theme coordination today, Jason.
Ooh,
Ooh. So I'm going to talk about a paper published in Nature Immunology online on the 30th of June. The article is called CD4 T cells lysen Kupffer cells to reverse CD8 T cell dysfunction induced by hepatocellular priming. So I guess that's pretty self-explanatory. First authors are Valentina Vencin and Christian Beccaria from the lab of Matteo Inacone at the San Rafael Scientific Institute in Milan, Italy. Nice place. And yeah, again, this is nice because I'm also going to be talking about kind of chronic conditions. So in this case, it's chronic hepatitis V virus infection. And yeah, some nice insight on how to improve the functionality of CD8 cells that are, the workhorse of the immune response against such a virus. And I think it's really interesting. It has a lot of potential for human therapy. So let's dive into it. So basically, chronic hepatitis B virus infection, big problem from some of the people that get this infection, and it's one of the characteristics of such a chronic infection is that usually you have some kind of dysfunctional immune response that is allowing this infection to stay, and this is a typical, I think, system of T cell exhaustion, dysfunctionality, and in this case, CD8 cells would be the ones that should be doing the work, killing infected cells and getting rid of the cells, and this is not what's happening. So in this work, they are looking into ways to restore CD8 activity, and so they're looking at a CD8 T cell best friend, who is a CD4 cell that is usually, in many cases, there to support the activation, and we know that CD4 help to CD8 cells is important in the context of many immune responses, and it looks like hepatitis B infection is not the exception. So they have a nice mouse model. They spend the first part of their work looking into generating a CD4 HBV specific CD4 T cell model. So basically, they have an infection, they have infected mouse, and they look for CD4 clones, and they identify TCRs that are responsive against a specific envelope protein of the virus, and they are able to generate transgenic mice expressing this T cell receptor in CD4 cells that are recognizing a envelope 120. So they call it envelope 126 CD4 cells because they're recognizing a peptide from the envelope protein starting at amino acid 126. And basically, they show that this is a specific response, specific CD4 cells that are specific against this viral protein. And they use these cells to show that if you have a situation where you have cell transfer, you have adoptive transfer, you can prevent you can kind of cure the chronic infection of the HPV by including these CD4 antigen specific CD4 cells, and this in a way allows the CD8 cells to become more activated and actually kill infected cells and clear the infection in a way that without the CD4 cells you don't see this. And so they have a few experiments which show that even if you have a delayed transfer, so first you have the CD8 cells and then a week later you put the CD4 cells, that still helps establish the CD8 response and reverse this dysfunction that is in CD8 cells. And And then when they start trying to understand how is this influence working, they show that CD4 cells are found mostly in the liver and not necessarily in secondary lymphoid organs. And this is another important part of our paradigm of how we understand T cell priming and T cell activation. We always think secondary lymphoid organs like lymph nodes are the place, or the spleen also in mice. I think is also very important. Is the place where T cells go to find the APCs and they interact with APCs, and also potentially the APCs are going to be interacting with both CD4s and CD8s to transfer this help signal from the CD4s to the CD8s. But then they start looking that there's a lot going on in the liver itself, not necessarily in the liver draining lymph node. So basically they do some imaging, and they see these triads of antigen presenting cells together with CD8s and CD4s together, and really indicating that there is some three-way communication or two-way communication from the APC, really indicating that this CD4 help is being relied on to the APC. And for this, you need MHC class 2 and a lot of model. But what I think is interesting is when they try to understand which is this APC that is licensing these CD8 cells with the help of CD4 cells, they show that it's actually not dendritic cells that are found in the liver. And they also do some studies in which they prevent trafficking of cells through the lymph nodes, and that does not affect the licensing of CD8 cells. So this really indicates that this interaction is happening in the liver and does not require a secondary lymphoid organ. So when they look in the liver, who is the cell? Which one is the cell that is doing this work? And first, we always think of dendritic cells, but they zero down on actually Kupffer cells, which are these resident kind of macrophage-like cells. And these seem to be, so they do depletion experiments in which they get rid of the dendritic cells. That does not affect the licensing, but if they get rid of the Kupffer cells, then they see a strong reduction in the activation of the CD8 cells. So they see a loss of this effect from the CD4 cells, really indicating that it's not the hepatic dendritic cells, but the Kupffer cells that are doing this work. And what's interesting is what exactly is the signal? So what they show indeed is that it's CD4 cells that get activated. They interact with Kupffer cells through CD40 and CD40 ligand interaction, which is a very established way of licensing the antigen presenting cells. And then it's production of cytokines from the Kupffer cells that is very important for the proper activation of the CD8 cells. And in particular, it's IL-27 production by Kupffer cells that seems to be the critical step that relies the interaction of the CD4 to the CD8 cells through the antigen presenting cells, to the point that if they inhibit, I think that they do antibodies against IL-27, they abrogate this activation, and they can actually imitate, they can get much of the effect of the CD4 cells by administering recombinant IL-27 alone. And that already reinvigorates the CD8 cells in the chronic infection as much as the CD4 cells would be. And interestingly, they don't see any kind of systemic toxicity, even though they are administering IL-27 systemically. And this is because the effect of IL-27 seems to be really required TCR signaling in order to activate the CD8 cells. So that's also interesting because they take some cells from some patients with chronic infection, HPV infection, and they show that by adding IL-27, they can increase the frequency of interferon gamma positive cells from the PBMCs of these patients, indicating that maybe also this could also be applied in humans, that maybe IL-27 could be an interesting therapeutic to help patients with chronic infection reactivate their CD8 cells and finally kind of get rid of these chronic infections. So really cool. I really like this paper. It was such a nice kind of step-by-step development of the story. And I think the therapeutic potential of IL-27, at least it seems really good. So I'm looking forward to see whether they're going to bring it to some clinical trial, something like that.
That would be interesting. And
Yeah.
cytokines are easy to make, so
Yeah.
you do it.
Yeah. So no, that's a really nice, really nice work.
That's pretty cool.
Yeah.
All right. Well, then, we're going to be speaking with Dr. Johnathan Canton at the University of Calgary in just a moment. But before we get that, do you want to differentiate human pluripotent stem cells into monocytes?
I do. No, actually, I don't. But sure, somebody does.
Somebody does. All right. Well, if you do, check out the STEM-Diff monocyte kit from STEMCELL Technologies. It generates millions of monocytes that are ready for downstream assays or further development into macrophages or dendritic cells. Learn more at stemcell.com/monocyte-kit.
So now for the second part of our show, for our interview, Jason and I are so happy to have with us Dr. Johnathan Canton. Johnathan is assistant professor at the University of Calgary, and his lab has really some very nice, interesting work done on phagocytes, and I think particularly dendritic cells and cross-presentation. So I think we're going to have a really, really nice discussion about these very powerful cells. So with us, Johnathan Canton, thank you so much for joining.
Thanks for having me.
Pleasure to have you here.
All right. So, I think macrophages and dendritic cells are great, and I think, of course, I'm mostly a T cell biologist, so I am biased, but I like to think I'm very open-minded, and I also care about all these other cells. And a lot of your work or most of your recent works working on dendritic cells, cross-presentation. So maybe would you like to give to our listeners a little primer, a little introduction on what are phagocytes, why are so important to study? I know this sounds kind of obvious, but from your perspective, why are you interested in these cells?
Phagocytes, yeah. So my interest in phagocytes actually started when I was in graduate school. So I was studying tropical parasitology, believe it or not, studying a parasite called Leishmania. And actually, they live inside of a phagocyte called macrophage, which you mentioned. And that really sort of opened my eyes to these really amazing cells. I mean, they don't just sit there. They're always moving around, engulfing massive amounts of solid material, of fluids, and really effectively processing it, metabolizing it. And I just found them to be incredibly captivating. And so from parasitology, I became interested in macrophages. And sort of towards the end of my first postdoc stint, I started getting more and more interested in dendritic cells because there weren't as many of them as macrophages, they weren't as abundant. But as soon as you took them out of the picture, you just seem to lose immune responses. And I found this incredibly interesting. And I wanted to know exactly what was special about dendritic cells. And so that sort of is what leapfrogged me into studying the biology of dendritic cells. Yeah.
So what is so special about dendritic cells?
Yeah, I mean, we often hear about them. If you've taken any sort of basic immunology course, we often hear them being referred to as sort of that bridge between the innate and adaptive arm of immunity. They're one of the first cells on scene. They sort of sense and sample material and then relay that information to the adaptive arm of the immune system. But oftentimes we hear that also in the context of other APCs like macrophages and whatnot. What really got me interested in dendritic cells was work coming out of many of those sort of labs that we've all heard about, Kenneth Murphy and some of these other labs, where they were actually effectively removing these cells from mice, and all of a sudden certain types of T cell responses just disappeared. And that really captured my attention because it implied non-redundancy. And that for me was like, okay, that seems like a really cool cell biological problem. What is it that these phagocytes are doing that others are not doing? And can I contribute to figuring that out? So yeah.
So some of your recent work already mentioned is looking into the phenomena called known as cross-presentation by dendritic cells. And I think it would be nice that maybe you also introduce this a bit. So our audience has, basic immunology, but what is our current understanding of the mechanisms of cross-presentation? What does cross-presentation really mean? And why is it important that some dendritic cells are able to cross-present?
Yeah, I think that's a really good question. And I think it's still an open question. I think it's a sort of a work in progress, right? So how does cross-presentation work? It's not new. We've known about it for a really long time. I think there have been like significant challenges in our understanding of cross-presentation. One of the main ones being dendritic cells are really hard to work with. They're hard to get in significant numbers. There are different types. Some are really not abundant. They die if you just look at them in the wrong way. And so I think there have been significant challenges to really understanding the biology of cross-presenting dendritic cells. But ultimately, um, what we're trying to understand is how they can take material up from outside. So, for example, the common example is how can a dendritic cell eat material that's outside of it, maybe a dead and dying cell that's lysed because of a viral infection, or maybe necrotic cells in the core of a tumor, and process them in such a way that they're presented on MHC, that peptides from them are presented on MHC1 to CD8 T cells. That question is still the topic of debate. And I don't think, I don't think we have an answer to it. So for me, this is a beautiful sort of black box in cell biology and presents a lot of opportunity for very interesting biology. So ultimately, what I would like to understand, and many other labs would like to understand, is how do cross-presenting dendritic cells process material to present to CD8 T cells at the cell biological level.
So I'm going to play dumb here for a second. What do you mean we don't know how? Don't they just phagocytosis, process it, and spit it out?
Yeah. So I think, I mean, that you get right to the crux of it. But so many other cells can do that, and yet they fail to generate CD8 T cell responses. So there's something different that's happening at the level of, for example, a conventional dendritic cell. We often talk of CDC1's, conventional dendritic cells type 1, as being the non-redundant cell and generating CD8 T cell responses. And we know that they're doing special things. So they are phagocytosing material, and they are processing it in much the same way that another phagocyte would process it. But they're also doing something unique. And there's one school of thought is that they're diverting cargo from within a phagosome to the cytosol of a cell. And if I asked you the question, well, how do they do that? Because what they're moving across those membranes are nominally membrane impermeant macromolecules, so either intact proteins or polypeptides. We actually don't know of any mechanism by which a cell can move polypeptides or proteins like that across intact phagosomal membranes. And so that's one question that we really, there's not a lot of clarity there, and there are multiple hypotheses, and that's one thing that we work on. And then there's another school of thought that that material can all be processed within the phagosome, loaded onto MHC1 in the phagosome, and then trafficked to the cell surface to be presented to a CD8 T cell. I don't know that it's one or the other. I think it may be context dependent. But that first question that I asked about how do you move things across phagosomal membranes, that's the part that I think is really interesting, and several labs are trying to figure out.
Because for those of us who might have forgotten the pathways, you don't have the pathways in their mind. So when we're thinking of classical loading of proteins on MHC plus one molecules, and classical, external loading of external proteins in MHC class 2. What are the characteristics of each path that we need to kind of keep in mind in order to understand why it's so puzzling that one is going to the other?
So typically we think of loading of MHC class 1 as something that we often call that the endogenous pathway of antigen presentation, right? So every nucleated cell has MHC1, and typically we get material in the cytosol, so viral proteins being processed by the proteasome into peptides that get imported into the endoplasmic reticulum and loaded onto MHC1 and trafficked to the cell surface. And then we think about MHC2 as separate. So we'd call that the exogenous pathway. So material comes in either through endocytosis, macropinocytosis, or phagocytosis, and winds up in a membrane-bound compartment. And then the digestion is not by the proteasome, but by proteases that exist within that endocytic organelle. And then we get loading onto MHC2 within that organelle, and that material gets trafficked to the cell surface. Where cross-presentation is different is we get exogenous material coming in to an endocytic organelle and making its way onto MHC1. So that's the distinction between the two. And so either some of that material makes its way into the cytosol and is processed as would happen in the endogenous pathway, or it might be loaded onto MHC1 within the endocytic organelle. So those would be the key differences between the pathways.
And so you're doing interesting microscopy to track what's happening, right? Could you talk a little bit about that and how you're solving what you're doing to solve the diffraction limit problem?
Yeah, so when we set up our lab, so we were I was very strategic about this, right? So I did 2 postdocs and initially trained in a really strong cell biology lab in Toronto. That was the lab of Sergio Grinstein. And he did really beautiful work on macrophages, predominantly on phagocytes and imaging techniques. We spoke before the podcast a little bit about single particle tracking, super resolution, correlative light electron microscopy. So I had a really strong background in applying state-of-the-art microscopy to cell biological questions in phagocytes. Towards the end of that first postdoc, I kind of came to the conclusion that I didn't want to start a lab to study macrophages or to do the cell biology of macrophages because there were just so many people doing such a beautiful job of that. And so I wanted to do something a little different. And that's when I went to do a second postdoc with Cayetano Raices Souza at the Francis Crick Institute, where I learned dendritic cell biology. And I got to start applying some of those cell biological and microscopy techniques now to a new cell, which was the CDC1s, for which there was very little actual cell biology because of all the challenges that I mentioned previously. They're very hard cells to work with. So that was sort of the start. And I decided when I started my lab in Calgary, I wanted to be the lab that was applying really strong cell biological techniques to CDC1s. I wanted to sort of fill that niche. And so when we got here, we started doing sort of what everyone else was doing to answer these questions, super resolution. So whether that's STED or different modalities of turf. But what we really wanted to start doing was a technique I had started learning at Cayetano's lab and really made my own here, which is correlative light electron microscopy, going from living dendritic cells, so imaging live dendritic cells, processing them, and then imaging them by a technique called focused ion beam scanning electron microscopy, or some people call it FibSEM or 3D EM, which takes thousands of slices through the very same cells that I image live on the light microscope, but it takes thousands of slices and then we do electron microscopy. So now all the things that were missing in the light microscopy, the black space, we now fill that gap with the 3D electron microscopy. We could see all the things that we couldn't see with the fluorescence. So that's a technique that we're applying every week now in our lab, and we're finding that it's really helping us to answer the biology of these CDC1s.
Maybe you would like to share with us in your latest, so you have a publication from last year in Science Immunology. Immunology, and in which you're looking into the role of certain apolipoproteins on phagosome rupture and antigen cross-presentation. And when I'm looking at this paper, as you mentioned, there's a lot of really nice microscopy images, and I see some EM images here. So I guess you really use this technique. Would you like to maybe walk us through a bit that publication and what are the insights that you got, and how this kind of very, high-tech techniques help you see these results in your systems?
Yeah, so I think when we were starting the lab about 4.5 years ago, there was a little bit of noise in the literature coming from the Raisa Souza lab, but also from many other labs like the lab of Sebastian Amigorina and others, which this notion was out there that maybe conventional dendritic cells, CDC1s, had what was being called leaky phagosomes. And this really goes back to a hypothesis that was first proposed in the 90s, actually, by Ron Germain and Cayetano Raisa Souza, called the indigestion hypothesis, which holds that in certain types of cells, certain types of dendritic cells, that the phagosomes may have unique properties that allows for material to effectively leak out of those phagosomes into the cytosol. Now, this gets back to what I was talking about earlier about having to get proteins out to be processed. But as you can imagine, that was a problem that, for decades was very, very difficult to solve, because how do you look for leaky phagosomes? What does that mean? Is that pores in the phagosomal membrane? Is that compromised membrane integrity or rupture of some sort? What is it? And if you're trying to find broken membranes in pictures, for example, microscopy, how do you know it's not an artifact of fixation? So when we started the lab, we wanted to sort of understand the machinery that was driving this so-called leakiness. And there was a lot of interest in it. And so we basically started out by looking for things that CDC1s, or the cells that have these leaky phagosomes, express that other cells do not express, and generating sort of a short list. And then we started doing our microscopy to see which of these things were present on a phagosome. And to our delight, we found that one protein called ApoL7C, it's an apolipoprotein in the L family, is not only uniquely expressed by CDC1s, we don't really find it in many other cells in our body. It's very unique to these cross-presenting cells. But to our delight, it was just lighting up phagosomes. Any phagocytic target that we fed to these cells, we saw this ApoL7C accumulating in large quantities on those phagosomes. Why was that so exciting for us? Because it turns out that these ApoLs have pore-forming domains that are known to disrupt biological membranes. And so the next question we asked was, well, what is a pore-forming protein doing on a phagosome in a dendritic cell? Well, the obvious answer is it's making them leaky. And so we started loading those phagosomes with all sorts of membrane impermeant dyes and proteins, and the answers were just crystal clear. When you had the recruitment of this ApoL to phagosomes, all this membrane impermeant material started leaking out. And so that was sort of huge for us, because it meant, wow, we got a cool story. We just started our lab and and we found something fun. But what really sort of sealed the deal for us was we generated a knockout mouse, and we found that these knockout mice, DCs from these knockout mice, had a severe defect in their ability to cross-present. And if we immunize those mice, they also had a strong deficiency in their ability to generate CD8 T cell responses. So that cell biology was translating into interesting in vivo phenotypes. And so, you started also by asking, well, what about the microscopy? Well, without the microscopy, we wouldn't have been able to see this discontinuity in the membranes. We wouldn't have been able to see all these membrane impermeant dyes leaking out. So these techniques really enabled this discovery in, I think, one mechanism by which DCs can move material to the cytosol.
So are you having to tag stuff then with EM to be able to know that that's the cargo of interest and everything and get down to the granular detail?
I really love your thinking. We haven't done that yet. So being able to see cargo move by EM is not something we've been able to do. The limit of what we've been able to do is sort of find quote unquote leaky phagosomes that have recruited our ApoL7C protein and then go in and look at the entirety of that phagosome and ask, are there obvious sites where the membrane topology looks different? Maybe it's been disrupted or there's a break in the membrane. So we've been able to do that to support all of our quantitative light microscopy. But I really do love your thinking about, well, can you actually detect the movement of material in and out of phagosomes? No, we haven't. We can only do that using light microscopy techniques so far, but it's something that we're definitely interested in.
So, changing a bit the topic here. It's very nice for us to ask our guests about their career, and especially if they have had, something out of the ordinary. And we were talking before we started recording that you are from Belize, this is a small country in Latin America. So, amigo, hermano latinoamericano. I'm from Argentina. And so, and you have a very interesting career story in which at some point you try your luck going back to Belize and opening an organic farm. And I was hoping you could just shortly share that experience with us and tell us what brought you back to your science. And I guess you just seem to have been the right answer, given that you're doing so well and have, doing so much interesting science in your lab.
Yeah, no, I love talking about that. It's one of my favorite things. Actually, that article it came on a request from Adrian Liston. So I was at the Canadian Society for Immunology, and I got to meet him there, and then he asked me to write it, and I was like, "Oh, really? You want to hear that story?" And it's been amazing that since I wrote it, I didn't expect anything from it, and so many students at conferences and postdocs have been coming up and saying, "Oh, I read that, and it meant so much." So now I actually do enjoy talking about it, but it comes from turmoil actually. So I like many of us that live in developing countries, we have to leave home to pursue our careers. In Belize country, population of 400,000, we have one university, and you can't get a master's degree in the sort of thing that I would like to study or a PhD. So from very early on, I knew I wanted to pursue a PhD in biology, but knew I would have to leave to do it, right? And that was a hard decision because I love Belize. I go back all the time. So, I left and I did my PhD and I did my postdoc, my first postdoc. So I was away for about 11 years. And I just was getting these recurring feelings of guilt. You're the brain drain. You left, you didn't come back. You could be applying those same skills here in Belize. So that was something that I think is not unique to me, but is fairly common to many people who leave their homes to go abroad and study. Around that time, I met my wife, and she was very interested in food politics, and we were talking about Belize a lot. And uh, yeah, I said, well, let me give this a shot. Let me take a break, fully feeling that if I go to Belize, I can never come back, because there's this narrative in science. Maybe it's just me, but I think it's fairly common for a lot of us that if you leave, your chances of coming back are pretty slim, because it looks like you took a break and you shouldn't have taken a break and you're not dedicated to the science and so on. So when I left, I thought I was leaving for good. And we went back and we did no science. I started an organic farm, as you say. And it was a wonderful experience. But I started treating the farm like I treat a lab. So I started saying, well, let me read the science on this, and let me try doing it like this, and let me ask questions. And at some point, my wife looked at me and said, don't you just want a lab? Isn't that sort of your your passion here? So again, great deal of turmoil. Well, how can I get back in if I want to do science again? They're not going to want me because I left and I went to Belize. Anyway, I started applying for positions, a second postdoc, and I applied for 3, and all 3 said yes. And my only interaction about having run a farm was, oh, wow, that's so cool. You went and did something different and ran a farm. And so it's one of these things that I think has for me beat a certain stigma that I had. And I often share that with my students that, hey, look, if you ever feel that you need to try something different, if you're unsure at this stage, just do it. You can always come back, and that was one of the big lessons for me. Importantly, I haven't stopped that farm. So I'm doing science here, but we got the farm to a point where it was running and it's still going. And so we still have that operating. And so I feel like I have the best of both worlds. I have something happening in Belize, which I'm very passionate about. And I have my science here, which I'm also very passionate about. And so, yeah, that's sort of that segue story.
All right, that's awesome. So usually at the end, we ask a follow-up question or something interesting about the person. I'm going to modify this because I don't want you to cheat. So normally it's if you weren't a scientist, what we would be. But it's if you weren't a scientist or an organic farmer, what would you be? Because I'm not letting you just say organic farmer after this one.
Yeah, you got me. Yeah, that's actually the typical answer that I would give.
All right, yeah, too easy.
Yeah, so if I weren't a scientist or if I weren't, I think I would be a fisherman. I grew up fishing.
Right.
And I love fishing, and you've been to Belize. I know that, so I know you understand. It's a fishing mecca, and it's one of my passions. So that would probably be it.
What's the biggest fish you've caught?
No, come on, that's not a good.
Oh, come on.
That's such an American question.
No,
What's the biggest fish?
These waters have really big fish around.
Maybe what's the most interesting fish? Or which is
interesting fish? Oh, that's a real question.
Yeah. Well, you know what? Like the biggest fish probably is a tarpon. I can't remember the exact size. It's maybe about hip height. But I don't do deep sea fishing, so I never really go for big fish. So typically small things.
Yeah, I was thinking the deep sea fishing out in Belize is wild.
Yes, it is. Yeah.
Well, but that's I have to say your story was very interesting. And it's funny that you say because now I am reaching the 11-year mark myself since I left Argentina, and yeah, it's I think the struggle is real, and what you mentioned really resonates with me. It's really tough knowing that I come from a small village in the mountains, also no place for a scientist working in immunology there for sure. And it's always tough to know, make that choice of going and, potentially never coming back. Most likely never coming back. So
Yeah. And I think it's something that a lot of people don't realize, but so many scientists are making this decision, right? And you look at how many people are coming from developing countries and doing science somewhere in a developed country, and you realize, wow, a lot of people are leaving a lot behind. And so, yeah.
Yeah. See, if that is not commitment, if that is not interest for your science, I don't know what is. So I guess that in comparison, taking a 2-year hiatus, it feels like very small. So
yeah.
Well, it was so nice talking to you, Johnathan. Thank you so much for joining us and talking about your science and your career. And I'm sure our listeners are going to be very happy from our conversation. And so, we had with us Dr. Johnathan Canton from the University of Calgary. Thank you again for joining us.
Thank you for having me. It was wonderful.
Thank you for being here. That brings us to the end of our show. Don't forget to subscribe to our newsletter at www.immunologypodcast.com to get the show notes, including an episode summary and links to all of the interview and Roundup papers. You can also reach out to us on X at @immunopodcast or via email info@immunologypodcast.com with feedback or suggest guests. See you next time.